For my term paper, I'm writing about the rabies virus on a molecular level and the current and past vaccine options. The article I've chosen to discuss today is called The Critical Role of K1685 and K1829 in the large protein of the rabies virus in viral pathogenesis and immune evasion. To introduce this subject, I'm going to first talk about the rabies virus. It is one of the oldest infectious diseases noted and still presents a large health risk in Asia and Africa. The L protein is believed to be essential for the rabies transcription and replication, and the L protein's role in viral pathogenesis and immune evasion was not known. The KDKE tetrad motif in the L protein is related to the methyltransferase, which is also called the MTase, activity in the viral mRNA process. In this study, a series of mutations in the motif were constructed with recombinant rabies virus mRNA. Both of the mutations were found to be stable, but unnaturally thin phenotypes were presented. They were more sensitive to expression of interferon-stimulated gene production, the IFIT2, than the parent virus. Both of these facts are important to its pathogenesis and immune evasion during infection. These findings improve our understanding of pathogenesis and are important for potential vaccine. The rabies virus belongs to the family Rhabdoviridae and belongs to the genus Lysovirius. The virus has a non-segmented negative strand RNA, which consists of a nucleoprotein, phosphoprotein, matrix protein, glycoprotein, and large protein. The large protein is an RNA-dependent RNA polymerase and is the discussion of this study. The rabies virus works through creating a ribonucleoprotein complex. This means that the RNA genome tightly creates a shell for the nucleoprotein, phosphoprotein, and large protein. The rabies virus made in labs is created to stimulate innate immune responses, but the street rabies virus evades immune responses, which makes it so dangerous. The reason it is able to hide from our immune systems is because of the restricted enhancement of the blood-brain barrier, and once it crosses the barrier, it causes mild inflammation and barely any neuronal destruction in the central nervous system. In this study, to test the street rabies virus, they put 6 cultures in plates infected with the rabies virus, and they removed the supernatants. They created a titration by direct fluorescent antibody assay, and the virus neutralizing antibody titers were measured during the test. What this really means is they were looking to see how many antibodies were reacting or not reacting to the rabies virus. Once that was confirmed, they lastly tested on mice. The results of this testing showed that the rabies virus has an unconventional capping mechanism and that very little is known about the L protein. There is no easy system for reconstructing transcription and replication, and the details of the replication mRNA modification is not clear. However, through this testing, we did find out why and how the street rabies virus is able to hide from our antibodies in our immune system, and that will help us create much better vaccines and possible post-infection treatments as well. I hope you guys have learned a little more about the rabies virus, especially the L protein, and the fact that it can hide from our immune systems, making it so dangerous. This is why all of our pets should be vaccinated so that we don't lose any furry friends.